nephron, as I told you, is the structural and functional unit of a kidney. So if you now look at this nephron, it is made up of one expanded end or beginning, what we call the glomerulus. This glomerulus consists of tuft of capillaries. So there are blood vessels that gets round, they form round around themselves, just like a ball. The blood capillaries, they twist and form a ball-like structure that enters into a cup-like, part of the nephron, what you call the Bowman's capsule. So the Bowman's capsule is harboring or containing this tuft of capillaries. So the combination of all this, the Bowman's capsule and the tuft of capillaries, is what we call glomerulus. And this tuft of capillaries now is made up of two vessels, what we call afferent capillary and efferent capillary. So the afferent capillaries brings blood into the renal tubule. The efferent capillary takes out blood out of this tuft of capillary to other part of the kidney. So now the blood is being brought into this tuft of capillaries, and so anything that is contained within that blood is going to be filtered through this Bowman's capsule. So the Bowman's capsule here, this area, is just like a sieve, area. So it is going to sieve out all the toxic waste, the urine, the water that is in excess, the urea from the protein metabolism, ammonia, and other, components of the blood that are not important to our own body. So all of them, they are going to be, sieved out of this blood capillaries into this Bowman's capsule. And so the Bowman's capsule now, it continues, it has a beginning, what you call the neck of the Bowman's capsule. And then from there now, there is a convolution of the tubule, what we call proximal convoluted tubule. So after the convolution, the tubule now descends down, and then it continues down, what we call descending part of the tubule, but this time around is thinner than this thick portion. So now this one loops inside this medulla of the kidney, in the medulla and the pyramid of the kidney and what have you. So it ascends up at the thin ascending of the loop, and then before it now continues into part of the distal convoluted tubule. So you can say that the nephron is made up of, tubules, descending and ascending, plus a loop. This loop is what we call loop of Henle. Loop of Henle. Henle is the name of a person. Just like you say loop of Annas, for example. So loop of Henle is a loop, part of the tubule, of a nephron. So we have seen that the nephron has Bowman's capsule, a proximal tubule, a distal tubule, and a loop. The proximal tubule has a convoluted portion, what we call proximal convoluted tubule, and proximal descending thick tubule, and then the descending thin tubule that forms part of the loop of Henle, and then you have ascending part of the loop of Henle, and then this one continues into the thick ascending distal tubule, which finally enters into the distal convoluted tubule, and this tubule now connects with what we call collecting tubule. This is what? Collecting tubule. This collecting tubule is the one that comes and get connected with the minor calyces, so that all the urine that is being formed from here, it enters here, gets around this portion, enters into the distal convoluted tubule before it finally enters into the collecting duct. And then it comes here, and so it gets out like that. And that is the different portion of the, nephron. And so each of the nephron, as I told you before, has a glomerulus that consists of the tuft of capillary and Bowman's capsule. So if you now take this portion of the glomerulus and you now look at the capillaries, if you can remember your own cardiovascular system, in each of a capillary, you have what you call an endothelium there. It's made up of a flat cells, flat squamous cells, So flat cells, they are the squamous cells, what we call endothelial cells. If you can remember, we have how many types of capillaries? 3. We have continuous capillaries, discontinuous capillaries, and then we have fenestrated capillaries. The type of capillaries that we have here are the fenestrated kind of capillaries to allow for filtration of urine from this blood into this Bowman's capsule. So in each of these our capillaries, each of the capillaries is been lined by this simple squamous epithelium, which is fenestrated. So there is a fenestration along the flat cell there to allow for the urine and other substances that are going to be excreted to pass into the Bowman's capsule. The Bowman's capsule, inside the Bowman's capsule now, I told you, is just like a cup-like structure, just like a funnel like that. So it is made up of 2 layers. Outer layer, which is closer to the glomerulus, this one, and then you have the inner layer. You get it? So you have the inner layer, which is this one, known as the visceral layer, and then you have the outer layer, which is the parietal layer. Each of these layers now is made up of, or is been lined by these single flat cells. So you have flat cells like this one. They are all lining this, Bowman's capsule there. So the Bowman's capsule in each area here is lined by this simple squamous epithelium. But immediately after the Bowman's capsule, you have the neck of the tubule. This neck of the tubule down to the distal convoluted tubule. These cells are been lined, these tubules, these areas are been lined by cuboidal epithelial cells. Single layer of cuboidal epithelial cells. But there are differences between the cuboidal cells that are found in the distal convoluted tubule and the cuboidal cells that are found in the distal convoluted tubule. So also they are different from the cuboidal cells that are found in the loop of Henle. The cuboidal cells that can be found right from the proximal convoluted tubule to the descending portion here, the cuboidal cells, if you look at it here, it has a centrally placed nucleus, right? It has a lot of mitochondria. Its own cytoplasm is eosinophilic. That means when you look at it under a microscope, it's red in color. That is what we call by eosinophilic, because it takes the eosin stain of that hematoxylin and eosin stain that you usually use when you want to stain in the laboratory. And if you look at the site of each of these cells now, it is a little bit, serrated, it's toothed, including the base. And if you look at the top, the apex of each of these cuboidal cells, it has some microvilli, just like fingers. So you can see that the apex of the cuboidal cells that are found in this portion is made up of this kind of cells, cuboidal cells, with centrally placed nucleus, multiple mitochondria, cytoplasm staining eosinophilic, serrated side of the cell membrane with the apex having or containing microvilli. Each of these cells now lies on the basement membrane. Are you clear? For the cuboidal cells that are found on the distal tubules, either the convoluted type or the thick ascending, It is made up of this kind of cell. It has a centrally located nucleus similar to this, has less of mitochondria as compared to this, looks less eosinophilic as compared to this, has few or no microvilli on the apex. Do you understand the differences? Good. I said it has a centrally located nucleus similar to this. It is also cuboidal like this. It has few microvilli as compared to this. It has less, or it looks less eosinophilic as compared to this. It has few or no microvilli in its apex. Are you clear? Good. It also rests on the basement membrane similar to this. You get it? So all of these tubules, all of this part of the tubule has been made up of this cuboidal epithelium, but single layer lying on basement membrane. But then these are the differences. Now, the collecting duct is the same thing. It's also lined by cuboidal epithelial cells, similar to that of the distal convoluted tubule and what have you. So this is just in a nutshell the epithelial lining the nephron, both in the proximal convoluted tubule, the distal convoluted tubule, the loop papilla, and in the collecting duct in a simple manner. So you can just draw these cuboids and compare the two, and then you make a reference to the proximal distal collecting and